Okay, hello. This is Dr. DiVincenzo. This is the first of your videos to help you kind of get through this tough time. These are basically supplementary videos to help you focus your studying. You're going to have to do a lot more learning on your own, obviously. Um, so from your reading and your slides that I post now are now complete sets of slides rather than ones that are missing material that we typically fill out in class. So between your slides and your reading in these videos, hopefully you'll get the information you need, and of course there's going to be obviously discussion and chats that you're going to be communicating with me with. Um, so this is the outline of your two sets. You got two sets of toxicology slides. This is the outline. And I want to say a couple things, as we move forward and point out a couple important areas. First, it's important for you to realize that not all man-made chemicals are harmful, and not all natural chemicals are safe. The periodic table obviously is all natural, what you call natural substances, elements. Some of those are very harmful, okay? And there are synthetic things that are completely harmless. So it's important for you to realize that. I gave you some information on on toxins and risk assessment, and I took you through some basic routes of exposure. Don't bother memorizing this stuff. Obviously, all your exams are still open book, open note. But I did want to talk to you a little bit about this. This is the slide that you might have seen on metabolic toxins. And I want to describe for you what the word heavy metals means. People use that kind of like very loosely. Because if you look at this list, I've highlighted arsenic in red. That's arsenic. And arsenic is not a metal, even. If you look at your periodic table, it's a metalloid. But these are called heavy metals. So it's kind of a general term that means metals and metalloids that are kind of toxic. But it also applies to the ones that are typically lower down on the periodic table, a little bit heavier, if you will. And there are different ways in which people define those in terms of like, the molar mass and everything in density, but that's not important for you to know. So something like sodium is not considered a heavy metal. Its molar mass, its number of protons is just too low. But things like lead, yes. Things like barium, yes. So it's just a real general term, and I wanted you to be aware of that. And one of the things that was talked about is the way these can interfere with enzymes by binding. So these particular species, like mercury and lead, they have an affinity for sulfur. It happens naturally in the environment, whether it be a lake or a soil or anything like that, but it also happens in your body. Well, it turns out sulfur is pretty important in enzymes. So what happens is they bind these metals to these active sites because they're attracted to the sulfur that you see there. And that actually changes the shape of the enzyme. And enzymes work on the idea of a lock and a key. You always draw, when we draw, I can't draw, but we draw pictures like this, and say, okay, yeah, one of these, this is the substrate, and this is the enzyme here, and it's kind of like they fit together, and that's how they work. That's how enzymes work. So if you change the shape, that has a huge effect, and that's one of the important things that was pointed out here. All right, so I take you through a little bit of the periodic table in some of the slides, just getting familiar. We've done some of that before, but I did take you through some basics now, gave you some definitions and elements and compounds. I talked about, I showed you a slide on all of matter and how it breaks down to, your elements, your compounds, your homogeneous and heterogeneous mixtures. And one thing I did mention there was chemical, or it's on the slide, chemical versus physical. It's important to realize that if I take something like a pot of water and boil it, that's a physical change. The water is going from liquid H2O to H2O vapor. It's still H2O. That's not a chemical change. It's just a change of state. That's a physical process. A chemical process needs to result in something new forming, basically. It could be a gas. It could be a precipitate. You often see like a change in color, things like that. So in order for me to separate the hydrogen from the oxygen, of course, water is a compound here. That requires a chemical process because I have to break these chemical bonds that are holding the atoms together. But to convert water from liquid to gas, these are just weak, what we call the bonds between the molecules, intermolecular bonds. rather than the bonds holding the atoms together. Those are easily broken in a physical process because there's usually enough energy there, whether it be heating a pot of water. But for something like this, like water here, to break those, that's going to be a chemical process. So if you look at these representations here, one of these figures is molecules of a compound, one is molecules of an element, one is a compound consisting of an array of ions, and an element consisting of an array of atoms. So if you look at molecules of a compound, well, compound is defined as having basically more than one element, right? And molecules would be these individual components. So C would have to be the correct answer here because you see that we have two different atoms making up this particular species. So those would be molecules of a compound, as opposed to molecules of an element, which would be A, because this would be like oxygen gas here. This could be like nitric oxide, which is one of the air pollutants we'll talk about later on. This is oxygen gas here. There would be molecules of an element. A compound, so you got more than one atom here, that would have to be D. This would be something like sodium chloride here. Those would be atoms in this array, or ions in this case. And then you have an element consisting of an array of atoms. This could be something like iron. Fe is iron. That's an E, not a C. Those would be the atoms in an array for an element. Okay, so it's important that you can kind of distinguish between those. A couple other things I talked about. Well, I already went through that, the physical and chemical stuff. So now here was a question I had asked on one of the slides. All chemical reactions occur at the same rate. Rate is just a fancy word for speed. And of course, hopefully, all of you answered false to that question. Of course, they don't occur at the same speed. They depend on several factors, and these factors are listed below. Now, temperature, an increase in temperature will increase the rate. Well, what happens at higher temperature? The species have more energy, so when they collide, they're more likely to have that energy needed to form product, if you will, because not all collisions result in reacting species forming product. So the collision has to be oriented the right way. The molecules have to be oriented the right way and have to have enough energy. So if I increase the temperature, I increase the energy, my chance of a successful collision leading to a product increases, and therefore my rate of my reaction increases. You can think about it the same way with concentration. The more you have, the more collisions you have, and the more likely that you're going to have successful collisions. Now, a catalyst is a species you add that speeds up a chemical reaction. And enzymes are examples of biological catalysts that catalyze most reactions in your body. And the key to a catalyst, too, is it can speed up a reaction, but it's not consumed. It basically leaves and then speeds up another reaction. So that's really important. And that happens in your body to a great extent. Most reactions are catalyzed by enzymes. The key is they're not consumed. They do that in several ways, just like that figure I showed you previously. They provide a site for species to come together, and that speeds up a reaction. That's where your catalytic converter works. It provides a surface for the gas molecules to come together and react. Now, this is important. Ions are really the active forms of metals. When I say active, I mean the ones that our body uses for nutrients, the ones that cause toxicological effects, the ones that form compounds. So when you think about something that's very simple like calcium, okay? When I think about building bone, it's the calcium ion that's used to build the bone because it's building a compound that is your bone. When I think about a nutrient, if you might take a calcium supplement because you're worried about osteoporosis, it might be in the form of calcium chloride. And your body would then, this would dissolve in your stomach, and your body would ultimately collect that calcium in the form of calcium 2 positive to use it. And if I have a substance that has a toxicological effect like mercury or lead, it's the cations that are typically much more dangerous than the elemental metals. The elemental metals are fairly non-reactive, and you can think of it this way. That lead wants to find something to react with to satisfy its positive 2 charge. It's not, it's not stable, if you will. It wants to be neutralized. So it finds a receptor in your body. It finds something else to replace. It might replace. This is something that happens with lead. It might replace calcium in your bones. That creates a structural problem that causes problems with your bones. So it's important that you realize the ions are the active forms. It's not to say that the element can't have any sort of toxic effect. Mercury vapor is somewhat toxic as the vapor. As the liquid, if you ingested it, it's still going to be toxic, but nothing like the mercury ion. And also, another thing we see is when some of these are bound to small carbon chains. So like methyl mercury, like this, very toxic. Having a small little carbon chain, a CH3 on it, makes it very toxic. These are some of the common charges that I showed you on your slides, and you get familiar with to some extent. You don't have to memorize these, of course. You have to memorize really anything in this course because you have open book, open note. Of course, the more you study, the better it prepares you. But I added chromium-6 because that's a pretty famous one. If you know the Aaron Brockovich story, that was hexavalent chromium that was causing that problem. That's the movie they made with Julia Roberts starring, but it's a true story about this town that was dealing with some toxic effects. They were seeing a lot of their kids and the citizens developing strange symptoms and things like that, and she was the one that was responsible for kind of identifying it and being the activist, if you will. Now I want to distinguish for you a couple things. Covalent compounds. Let's say that ionic compounds are formed from metals and nonmetals. An example I give you is calcium carbonate. So calcium's on the left side of the table. That's a metal. Carbon's a nonmetal, and oxygen's a nonmetal. This actually will, in solution, do something like this. Will come apart into 2 ions. But anyway, that is an ionic compound. Covalent compounds are formed from nonmetals only. So if you look at this here, this is basically glucose. You see C6H12O6. These are all nonmetals. These covalent compounds are the building blocks in biological systems. So when you look at proteins, carbohydrates, fats, and all that stuff, that's all built from these molecular covalent compounds that are made from nonmetals. And organic compounds are simply covalent compounds containing carbon. So those are the real biological building blocks. The exceptions to that are carbon dioxide gases. Those are considered inorganic. The next thing I want to point out is a difficult concept that was in your slides, and it's not in your book really, is these dot symbols for the elements. What it is, the group number is the number of valence electrons, and I think that I mentioned this before. The group numbers in the number of valence electrons, and you remember the old system is, 1, 2, 3, 4, 5, 6, 7, and 8. Well, you can just subtract 10 on this system. It's not a big deal. That's still 13 minus 10 is 3. This is the number of valence electrons. So everything in group 4 has 4 valence electrons. Okay? And the unpaired dots tells you the bonding capacity. So this is why carbon is, if you will, the building block of biological materials, because it has the ability to be a great central atom and form 4 bonds. So it can combine with a lot of things. Where something like hydrogen, well, it's not going to be very good as a structural atom because it only can form 1 bond. So, of course, carbon can bond to something here, 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 and here. So this is the number of valence electrons, and it is an indication, the group number is the number of valence electrons, and the single dots is an indication of the bonding capacity. So something like nitrogen would only be able to form 3 bonds. And these are generalizations, as always. There's always exceptions to these things. Everything we do in this class is really a generalization. Okay, And then as you go through the slides, I start to talk about things like neurotoxins, and we talk about an LD50, and things like that, some nerve agents. And ultimately, I get to something like this, the nerve transmission, what happens with some of these neurotoxins. So what you're looking at here, if you take your biology, you're looking at two nerve cells here. And this down here is a blow-up of the image up here, close-up of that. So here's kind of your cell body with your dendrites and all that, and your axon, if you remember this stuff. At the very end here, we've got these neurotransmitters that are released from these vesicles into the synaptic gap. And then the next cell next to it has the receptor sites. And you can see very there, much like the enzyme, it's a lock and key type mechanism, and they fit in there. Well, what some of these nerve agents do, these neurotoxins, is they interfere with this process. And and this is mainly insecticides because it's the nerve response here. So this is not going to be obviously in plants. The insecticide interferes with the normal neurotransmitter, which is acetylcholine. And basically, what it does is it attacks the enzyme and keeps the enzyme from breaking down the acetylcholine, which is this, which keeps collecting in the gap. This creates an uncontrolled response by the organism, ultimately leads to paralysis and death. And a toxin's LD50 is a measure of this, and I talked about that, you saw that in the slides. And that is, the lower the LD50, the more toxic. If the LD50 is 50 milligrams per kilogram, right, versus 100 milligrams per kilogram, that is the dose it takes to kill 50% of the population. So this one has a smaller dose and therefore is more toxic. No one get confused by that. You see the higher dose, you want to say, oh, it's more toxic. No, it's the exact opposite reasoning. Then there's a whole section on endocrine toxins. Endocrine toxins are really big now in terms of being studied. And these are compounds that kind of interfere with kind of our endocrine system. So things like estrogens. These are groups of hormones in animals responsible for things like feminization, and that is characteristics in reproduction. So that's just one of these endocrines, one of these hormones that produced by the endocrine system, and that these endocrine toxins can kind of mimic and interfere with. So what happens is sometimes they behave like these hormones, and they trigger a response in your body when it shouldn't happen. And that can happen a lot during puberty and stuff like that. So you wind up with some odd effects that are occurring in the environment. You see things like organisms with multiple genitalia, both male and female. You actually see reduced genitalia production and size and things like that. So all sorts of weird things happen. And there's lots of chemicals that we're using now in our products, and sometimes they're just basic products too, that are winding up in our groundwater and winding up in our drinking water ultimately. And I think one of the last things I want to talk about is carcinogens, and I want you to be aware of that there are natural things that are carcinogens. Peanuts have a carcinogen in them. Of course, you'd have to eat tons of them to ever get affected by that, but it's important that you realize that. These are not just man-made. But basically, they're chemicals that cause kind of uncontrolled cell growth and loss of differentiation of cells and things like that, kind of grow out of control. Most carcinogens are mutagens, and that is they cause a mutation in genes, but not all of them. Most mutagens are carcinogens, but not all of them. And viruses can be mutagens. They can insert DNA, right? I mean, a virus has an RNA genetic makeup to it. We're talking about coronavirus now. That is their nucleic acid. It's RNA. That goes in and kind of takes over the machinery of the cell. Of course, the coronavirus is not carcinogenic. But Zika virus is a teratogen. And that is, it will affect kind of the reproductive process in organisms. So teratogens are basically induced birth defects by affecting and developing embryo. So the Zika virus is a teratogen. Okay, and then this pulls us into the second set of slides, which is measuring toxicity. There's not as much here I'm covering on my video, but I want to show you this curve here because this is a dose-response curve. And what you have is dose on the x-axis and the response on the y-axis. That response could be death, or it could be just something like decrease in reproduction. It could be lots of things you can measure. And you can see by the curve, obviously, this would be 100% up here, response. And you have some sort of threshold dose. What that means is, first let me say that you have to realize this is generated by a bunch of data points. So it's not a curve. It's a curve fit to data points. So there's individual data points here. Okay, let's say those are our individual data points. Well, a threshold dose is the level below which there's no observable effect. So I would say, obviously, in this figure here, it's actually probably this point here, right here. Below that, there's no observable effect, so we call that our threshold dose. There's also the no observed effect level. It's the highest dose that does not show a response. That, if you will, would be this guy right here. This doesn't show a response. We'd say that one is actually the NOEL, and the threshold dose really would be this blue one here. So I had originally not drawn the data points on the original figures, so I just wanted you to distinguish between those. And this is usually done in dosage of milligram per kilogram, of course. Now this was a slide to help you kind of understand how we calculate and use this LD50. So the example I gave you, and this is my last slide, I think, or biomagnification is determining how many cups of coffee you would have to drink. Okay, so we have this 150-pound person, and of course I would give you this conversion, and this is the body weight conversion. And what we've done is convert this person's body weight to kilograms because we have a dosage up here, and this is for rats, but we're going to assume it's the same for humans. That's what it says, assuming it's the same for humans. Always look at your units. So this is the weight of my person now, and to figure out how much I need to kill 50% of the population, well, I'll multiply it by the LD50, and you notice I'll get the dosage in milligrams, right? Because my kilograms cancel out, so it's per kilogram of person times per kilogram mass of person times 68.2 kilogram person. Gives me this number of milligrams. And a cup of coffee has about 100 milligrams, so if we divide by 100, I do it as multiplying by the reciprocal, 1 over 100, you get 88.6 cups. And of course, if you're drinking 88.6 cups of coffee a day, you've got bigger problems than worrying about the LD50. And the last thing I want to talk about is biomagnification. This is an important topic, and it's something worth spending a little bit of time on. So some of these chemicals I talked about like dissolving like. If something's very water soluble, it likes water, it's not going to biomagnify. The ones that biomagnify are the ones that bioaccumulate in your fatty tissues. So something like DDT that doesn't like water, it's not very water soluble, it will collect in the fats of your body. If we analyzed, they tell you when you eat meat and stuff like that, if you cut away the fatty tissues, you actually expose yourself less to these chemicals. And if I was to analyze your body, most of the DDT in your body, which you do have, it's been estimated that every living organism on the planet has DDT in their body, I would find it in the fatty tissues. But what happens is it's stored in the fatty tissues and it doesn't break down. So here it is now, it's in these guys here, and it doesn't break down, and then a small fish eats a bunch of these smaller ones down here, these zooplankton, and now it's collecting DDT. And as it's breaking down that material for energy on the zooplankton, the the DDT doesn't get broken down, it gets stored in its fat tissue. So now these guys here have more DDT in their fat than these guys, and so forth. And this is the problem that resulted in birds of prey. And it wasn't just bald eagles, they were the poster child, but osprey and things like that were all affected by this because DDT interferes with calcium metabolism. It's a neurotoxin. We use it to fight insects like mosquitoes, which spread malaria. We don't use it in this country anymore. It's been banned in this country, but it's now used in some of your developing countries like Africa and stuff like that. That's a continent, obviously. But a lot of countries in Africa use it to fight mosquitoes because malaria is a real concern there. But it was banned because it was causing this eggshell thinning. We used it as a neurotoxin to kill insects, but one of the side effects, one of the other things it does in these bodies of these organisms, it interferes with calcium metabolism. That was the real problem. So it's important to realize that these chemicals, they can often do things in organisms that is not what their target role was. And that's one of the big concerns with chemicals in the environment and toxicology in general. So I hope this video was helpful to you, and I'll be making more of these, and you'll be watching more of these. So good luck with your studying.